The next topic in our syllabus is nucleosome. We have seen so far the structures, we have seen the initiation of it, that how the genetic material is there. We have seen it's a chromosome and it plays important role. Now next is the whatsoever DNA is there, that is basically wrapped, because we have seen, if you remember, while earlier one, when we have seen about the cell and various things. At that time we have seen that the DNA is very long, otherwise it is almost in kilometers, and if it has to be put into a small cell of micron size, then it has to be compacted very well. So this is one of the first important step in the compaction of this DNA. So it is basically the nucleosome is the basic structural unit of DNA packaging. So it is packaged in particularly is in eukaryotes. The structure of a nucleosome consists of segment of DNA which is wound around 8 histone proteins. There are 8 histone proteins are there, and they are actually basically 4 histone proteins are there. They are in duplicate. If you can see this figure, you can see these various histones, that is H2B, H3, H2A. The color is same, you can see here, and like this this color combinations are there. So these are called as a core histones. These are basically 4 there, that is H2A, H2B, H3 and H4. So these 4 are there, and it is compacted around these histones. So this wrapped around a set of 8 proteins, and therefore it is called as a histone octamer, because there are 8 basically protein. It is 2 copies each of H2A, H2B and H3 and H4. So it must be compacted in nucleosome to fit within the cell nucleus. That we have already discussed, that this compaction is important, and this is particularly compacted in such a way that the long molecule is compacted like that. It is wound around this or it is wrapped around these histone molecules, and then it makes a typical structure that is called as a beads around strings. So that is very popularly called as a strings and beads type of a structure, which is very, very important. In addition to that nucleosome, the eukaryotic chromatin is further compacted by being folded into series of more complex structure, and then it is forming as a chromosome lastly. Each human cell is containing about 30 million nucleosomes. You can imagine the number of nucleosomes. Each cell, there are around 300 trillion cells in human body, and each cell containing 30 million nucleosomes. Now you can understand the deepness or the depth of things which are available over here. So nucleosomes are thought to carry epigenetically inherited information in the form of covalent modification in their core histones. We are anyway going to discuss something about histones in our subsequent sections. The nucleosome position in the genome are not random, and it is important to know where each nucleosome is located, because it determines the accessibility. So this is very important in what structure is. Suppose particular enzyme has to be synthesized. Okay, for that enzyme that much RNA has to be expressed. So that much transcription has to taking place. So it should be located. For that, whole chromosome cannot be made into transcription. So only that much portion which is responsible for coding of that particular enzyme or that particularly protein, that is important. So that locations are becoming easy because of this kind of structure. I hope you are understanding. And this is the accessibility of the DNA to the regulatory proteins. So these regulatory proteins will access it, and accordingly only the required portion will be expressed, and not everything in that will be expressed. So this is what importance is. Will not go into the history of it. This nucleosome core particle consists of approximately 146 base pairs. Okay, slightly here or there. They are wrapped into 1.67 left-handed superhelical turns, and there are such superhelical turns are there. Superhelical means simple is a helix, and apart from helix, there is one more turns are there. During helix also there are turn, so we call it as a DNA as a helical structure. And in addition to that, there is a superhelical turns around the histone octamer. So these are consisting of two copies each of these we have already seen about H2A, H2B, H3 and H4. These core particles are connected by the stretcher of a linker DNA. So this is a linker DNA. In between two molecules, there is a tail-like a structure, that is a linker DNA. So this is a bead. There are two beads, and in these two beads there is a structure of without or unwrapped structure. So that is a linker DNA, and it can be around up to 8 base pair long. Technically, a nucleosome is defined as a core particle plus one of these linker region, either pre or post, like that. So this linker region, however, the word is often synonymous with the core particle only. Genome-wide nucleosome positioning maps are now available for many model organisms, including mouse, liver and brain. So now we are just like I told you, genome projects were carried out. So we have got genome project, and now we know about that where which kind of nucleosomes are present and for what functional activity these are there. So similar maps are available that in particularly this kind of chromosomes, these nucleosomes are present, and in this nucleosome what kind of activities are there. Particularly these are available for mouse liver and mouse brain. The linker histones such as H1 and its isoform are involved in chromatin compaction, and they are sitting at the base of the nucleosome near the DNA entry and exit binding to the linker region. And non-condensation nucleosomes without the linker histones resemble the bits on the string of the DNA, as we have seen. We have seen it is there, and it is found under electron microscope. The other in other cells like sperm cells, they use the protamines to package their genomic DNA, and they achieve even higher packaging ratio because they are still smaller cells and they are carrying a single unit of it. So they are there. Histone equivalents and simple chromatin structure are also been found in various other species, and it suggests that eukaryotes are not only the organism that uses nucleosomes, only the what we can structure differs. Now this earlier structure gives some kind of historical or research background that how it has been there. Let us go ahead. That is important, is that is nucleosome core particle. The nucleosome's core particle consists of about 146 base pair of the DNA. So this is how the wrapping takes place, and around it the DNAs are there, which are these are four color you can see: red, green, dark blue, so yellow. These are four colors: red, yellow, green and blue. These are four histone molecules, H2A, H2B and H3, H4. So these are the four colors there, and around that the DNAs are wrapped around each, and like that, and then it extends. So this is what called as a core molecule. So this is the core particle here. That figure is there, and this core molecule or core particle that consists of around 146 base pair. Already we have seen it, superhelical turns, and they are binding, etc., and there is a linker DNA of it. They are observed when the chromatin in the interface and it is treated. This is how the research has done. See, this is one of the technique. I will not go into these details. That basically the partial DNAs are there. DNAs means which will cause hydrolysis of DNA. So these DNAs will cause digestion of chromatin, that it will make the breaks, and these breaks will be how, particularly where the linkers are there, because the particle which are bound to the histone molecules, that may not be breaking, and therefore it will show particularly areas of that areas, that is 180, 360, 440 base pairs, because that is breaking at a, we have seen around 146 base pairs are there which are around that and plus few. So like that around 180, and that will be having equal everywhere. So this is having the equal length, it will show. This is called as a this blotting technology or electrophoresis. Okay, this you can see the figure which is writing apoptotic DNA laddering. So this DNA laddering is basically we have seen last time various technologies. So this is electrophoresis is one technology that causes separation on the basis of the molecular weight of this compound, and accordingly it will be there. So here if it is a simple DNA, it will be having this kind of bands, means some are here, like that. But here you will find that most of these are concentrated at one place, and that are more than 2000 base pairs. That means, so these are particularly reaching to at what place. So this is how it is there. So here it is digested one. So we are having a similar band of around particular size, while here it is not of particular size. So this is how we get the DNA. These are called as apoptotic DNA laddering, and this DNA ladder means ladder-like structure, CD-like structure. So this CD-like structure is there. So this kind of structure is called as a DNA ladders. How this are formed? So this is again another figure that shows the octamer of the core histone. So these are the 4 molecules, and this next portion is, we'll call it as a linker DNA. So these are linker DNA. Then the red color one is called as a core DNA. It is wrapped around, and there are 4 histones which are there, which are working over there. So what we have seen that these are the various structures, and these are the schematic representation. So these are not like that there. So this is the schematic representation, and this is the particularly electronic microscope structure that shows this kind of wrapping and this binding. So there are interactions between histone and DNA. The nucleosomes contain over 120 or more direct protein to DNA interactions. Because of this interaction only, that binding is possible, or it is not getting sliding down. Otherwise the band like structure may slide down. So these are there because of this bindings are there, and there are several hundred water mediated interactions are there. Direct protein DNA interactions are not spread evenly about the octamer surface, but they are located at the discrete sites, and these are due to the formation of 2 types of DNA binding site within the octamer. Alpha 1 site, which uses the alpha 1 helix from the 2 adjacent histones, and L1 L2 site, that is at every loop, that is whenever there is a fold, that loop is there. So this salt links and hydrogen bonding, these all hydrogen bonding, water this what we can say hydrostatic binding, van der Waals bonding, you know about it in chemistry. So these are various bindings are there, and these bindings are playing important role. So these are binding factors are there. They prefer some DNA sequence over the others, and they are capable of binding practically to any sequence otherwise. It is because of the flexibility of the formation of these water mediator interactions. There are non-polar interaction also, and they are made between protein side chains and deoxyribose group, and also arginine side chain intercalates into the DNA minor group. That all 14 sites they are having similar octamer surfaces. So the distribution and the strain DNA binding octamer, that will cause distortion of DNA within the nucleosome, and the DNA is non-uniformly bent and also contain twist defects. So these are the things which are happening. Then at histone there will be tails. So that histone tails domain will be there. They constitute about 30% of the mass of the histones. They are not visible in the crystal structure of the nucleosome because they are highly intrinsic flexibility. They have been thought to be largely unstructured, and the end terminal tails of the histone H3 and H2B pass through a channel formed through the minor grooves in two DNA strands, and it comes out or protrudes from DNA every 20 base pair. So this is what the DNA tail is. The interaction is having under physiological condition, and it suggests the acetylation of the H4 tail distort the high order structure of the chromatin. So it is important in the high order structure of the chromatin. What is this high order structure of the chromatin? That when it go on compacting one after another, so these are various models, they are expressed that it shows that in what form it must be compacted. So there are various ways are there, DNA, and how this DNA is compacted at various places, which are resulting into this kind of, lastly you will find that this kind of chromosomes which is there, which we call it as a X-shaped chromosome. Okay. So this compaction model, that is because of the high order arrangement mechanism. So this organization of DNA that is achieved by a nucleosome that cannot fully explain the packaging of DNA which is observed in the cell nucleus. We have seen at multiple pole it is there that so big DNA is compacted in a very small nucleus. The further compaction of chromatin into cell nucleus is necessary, but the exact mechanism behind that it is not understood. It is believed that the current understanding is that the repeating nucleosome with the intervening linker DNA form 10 nanometer fiber, and it is described as a beads on a string, because this 10 nanometer fibers is there, on that the beads means histone molecules are there, and they have packaging ratio of about 5 to 10. Then the chain of nucleosome can be arranged into 10 nanometer fibers. We have seen, I hope, in earlier one presentation I have shown you that this is there, and this is having packaging ratio of around 50, and then it will depend on the presence of H1 type of histone, and then lastly the crystal structure of this is presented, and it is used to build a proposed structure of 30 nanometer fibers, which is making 2 start helix. And there is still certain amount of contention regarding, there are various problems there because it does not show in the electronic microscopic data. So electronic microscopically it shows something different, but these are various beliefs that how this must be happening, how the compaction must be happening. So then there is a important thing that although it is a very stable protein DNA complex, it is not static. It is not static means it doesn't mean that it will remain as such. Suppose we have seen that around 146 base pair are wrapped, that it doesn't mean that only that 146 are wrapped as such. It may rotate, just like top है भौरा है तुम्हारे पास, और भौरे के चारों बाजू में आपने वो थ्रेड यू आर रैप्ड है ना, तो अगर वो भौरा घूमता है तो मे बी व्हाट विल हैपन, द थ्रेड विल आल्सो मूव, है ना. So at some places it will more, at some places it will be less. So accordingly it can be changed. So it is what we call it as a dynamic. It is not a static, and it can undergo number of different structural rearrangements including nucleosome sliding and DNA site exposure. So when a requirement is there, the nucleosome can make change in the winding. That means winding will remain same, but it may move here or there, and depending on the context they can inhibit or facilitate the transcription factor binding. The nucleosome position are controlled by 3 major contributors, that is intrinsic binding affinity of the histone octamer, that will depend on the DNA sequence. Second, the nucleosome can be displaced or it can be recruited by the competitive or cooperative binding of other protein factors. And third, nucleosome may be actively translocated by ATP dependent remodeling complex. So because of this thing, it doesn't remain static, it becomes a dynamic, it can move, it can change. And this is what importance is, because when particular transcription is required, particular area is required, and then it becomes easy that only that much area is there, so that becomes very easy to what part has to be taken out. So this nucleosome sliding, I told you that there can be sliding of that, there can be changes, slight changes will be taking place, and that will cause the exposure in the DNA site. I'll not go into the detailed studies, or rather it's not expected why it should be made into detail, but there will be, the movement will be there to what we call as a nucleosome sliding. Then it will result into DNA site exposure, that this DNA will be wrapped or unwrapped, always there is a equilibrium, as I told you, and this is sliding causes it. So the DNA which was wrapped earlier, that can become unwrapped, and that which was unwrapped earlier, that can become wrapped. So this is happening, and because of that the wrapped structure is not exposed, but the unwrapped is exposed, and accordingly this wrapping unwrapping continues. The DNA does not need to be actively dissociated from nucleosome, but there is a significant fraction of time during which it is fully accessible, and this is important, that this we say that the DNA within the nucleosome to breathe has important functional consequence. Okay. Breathe means which it becomes free from that. So dynamic breathing of nucleosome plays important role in restricting advancement of RNA polymerase due to transcription elongation. So this is how it can stop, or it can restrict it up to certain length of DNA only. There is an area that will be nucleosome free region, and this nucleosome free region will be allowing the promoter for the DNA accessibility to various proteins like transcription factors. Nucleosome free regions typically spans for 200 nucleotides, in particular that Streptomyces cerevisiae microorganism, where it has been studied. Well positioned nucleosome from the boundaries of this nucleosome free region can be seen. They are called as a +1 or -1 nucleosomes, and are located at the canonical distance downstream and upstream. From the transcription site, downstream means next to that, upstream means before it, and they are tend to incorporate in the histone, that is H2A, which is a Z histone variant. So these are various things which are there. We shall be studying histone later. We shall be studying about post-translational modification. They are important. We shall see that how this post-translational modification are a very important reason or basis for some of the problems which are there. So we shall study about it later. This whole thing is there in your next. What do you mean by acetylation and all? So we shall be studying this histone acetylation and various variants of histone in the next. There is a remodeling can takes place in the nucleosome. That nucleosome can be having structurally different structure. So these are there, and there are consequences for that. It can make the changes in overall development. It can cause any developmental changes. Again, maybe problems in the transcriptional factors. So this is the remodeling is there, that causes the enzyme. Remodeling always means a change in the structure briefly. So this is how this remodeling is there, and it is because of certain enzymes. In vitro, we do not to go for this detail. These are the scientific studies which are performed, how they are there, and what are various cross linkings are there with the core particle. So it has found that there are disulfide linkage. Disulfide you must be knowing in enzymes, or it is in coenzymes. There SH molecules are there. We call it as sulfhydryl group, and so when we so these are the thing. Disulfide, when SHHS group are there, when two S group are there, their example, there is a cysteine like amino acid, and there are two cysteine molecules are there, they are attached to each other, then it becomes a disulfide. So S to S, sulfur to sulfur, when this type of amino acids are there, then we call it as a disulfide ring. I hope you might have studied it in biochemistry. So or these are the sulfhydryl group containing amino acid, and when one hydrogen is removed, then it becomes, there are many coenzymes which are there with sulfhydryl group. So SH group or thiol group it is also called as a. So these are the important groups, and they are responsible for this cross linking, that is disulfide cross linking in the nucleosome core particles. So the two different cross link can be introduced into the nucleosome core particle. The first one cross link, the two copies of H2A via induced cysteine. I told you cysteine is one of the such amino acid. There are many, there are few amino acids which are having this sulfhydryl group, but cysteine is prominent in that, and it causes the histone octamer which is stable against the H2A H2B dimer loss during nucleosome reconstitution. Another cross link can be introduced between H3 N terminal histone tail and nucleosome DNA ends via incorporated through convertible nucleotides. So they are the these cross links which causes stabilization of the nucleosome against the DNA dissociation at very low particle concentration and at elevated salt carbon. Because of salt also there are the changes, and these can be prevented because of that. So these are there. We shall see all these proteins, that is H3, H4, H2A, H2B. We shall see about this in our next section about histones. So how these histones are, and these are the crystal structure, these are the specific things about it. So these are the important structures, these are the important, just a minute.